Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello, hi everyone. Alright, so we're going to have our first experiment today, which is a basic technique in microscopy. Alright, under this lab, we're going to get familiar with the correct technique and also usage of the microscope. Alright, so this lab consists of two subtopic, which is exercise 1.1 and exercise 1.2. Under 1.1, they got four, which is A, B, C, and also D. A. Calculating actual magnification of a light microscope. They are given the formula here. A total magnification is equal to the eyepiece length, or known as the ocular length, times with the objective length. Where is the eyepiece or ocular lens? Eyepiece and ocular lens is located right here. The objective lens is here. So normally in lab there are several types of lens, which is objective lens, 4 times, 10 times, 40s, and also 100. The ocular lens or eyepiece lens, they have several types also, 5, 10, and also 50. Basically, the higher magnification, the larger the image that we can get. And we can do this exercise by multiple these objective lens and also the ocular lens. So right here, we can have 100, which is by using an oil immersions. Alright, we can have 1500, which is the highest number of the total magnification. The lowest number can be as 20. Alright, so basically just complete this table in the lab report. B. Focusing and understanding a microscope image. Take a prepared slide of the letter A and we need to observe under 2 magnification, 4 times and 10 times. Normally, when we want to observe any specimen, we need to start with the low magnification first. For example, 4 times. Get the first image by adjusting the coarse knob. Once we get the first image, then refine the image by using the fine knob. Once you get a clear and sharp image, turn the objective knob into a 10. Normally, the image will get a little blur. Do not touch the coarse knob, using the fine knob to adjust the image. Then after that, we should draw the letter A, what we see on the microscope. So normally under lower magnification, we can see the whole letter A. Okay, then the E is a bit smaller compared to the higher magnification. So under higher magnification, for example 10 times 10, we can get a larger E, which is almost full view of this microscope. C. Field of view. This experiment is using the prepared slide, which is graph paper. Graph paper given here is normally is a 1 cm. Under 1 cm of the graph paper, we have 10 smaller boxes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1 cm equal to 10 millimeter. So we got here, right here, 10 smaller boxes also. Then we should observe this graph paper under 3 magnification, 4, 10, and also 40. Under 4 times, we can see that the graph papers is we have 4. 1, 2, 3, 4. We need to count the diameter of this field of view. So normally we can get 4 smaller boxes, which is equal to 4 mm. Then we move to the magnification 10 by using the same graph paper. So under 10 times, we can see that the image is getting larger, meaning that we can get a higher magnification the larger the image, but the diameter is getting smaller, meaning that we can see that they have only a diameter of one full box and half. So normally under 10 times we can have 1.5 mm because it's a 1.5 box of the under this image. Under 40, we can see that the image of the graph paper is getting more clear. We can see that here is the texture of the paper or the ink of the green here. So make sure that we adjust this one to the edges of the green one, and we have to count the diameter, how many boxes that we have under 40. Alright, so unfortunately, so we cannot see any one boxes here, and then we have to move this what we call the stage to the right or to the left until we get another green one. Alright, so we have to count how many view that we have. Alright, for example, under mine, okay, we have 3 circle. So we know that one smaller boxes here is equal to 1 mm. So I need to move by 3 times. So 1 mm divided by 3. So we know that one of the view in the magnification under 40 is around 0.33 mm. Alright, as a conclusion, we can say that the graph paper given is a 1 cm or equal to the 10 mm because they have 10 smaller boxes here. Under 4 times or low magnification, we can have a diameter of 4, 1, 2, 3, 4 or 4.5, for example. So the higher magnification, for example 10 times, okay, they go to the 1.5 mm. Alright, and then the higher magnification, we can get at least 1/3 of the diameter is around 1/3 from the 1 mm or around 0.33. So what we can say that is actually the higher magnification that we have, the smaller field of view that we have. Okay, so by comparing the size or the measurement of the diameter that we have just now. Okay, so as a result, we should draw the image. Okay, for example, like this 1, 2, 3, 4. Okay, so make sure you do in the correct way. Alright, so under 10 times, okay, the image of this one is a bit thicker. Okay, so the green one is a bit thicker. Alright, you can have here. Alright, and then okay, for example here. Alright, to make sure that they are thicker compared to the 4 times. Okay. Alright, what about the 40? Okay, so 40 you just draw something like this. Okay, so you can see that they have something like, is a crystalline, it's actually the texture of the paper graph. Alright, please complete the table in the measurement. Okay, diameter, field of view for the 4 times, 10 times and 40 objective lens. Alright, the depth of field using a cross thread, which is a different color of thread, and will be crossing at the center. So basically, the depth of field is a portion of specimen that appears sharply at one's image or at one view. So if the specimen is still thick, okay, and if we want to focus on the upper one, the lower one or the lower cell or the lower specimen will be out of focus. And then when we want to focus on the lower one, the upper one will be out of focus. Let's see under the thread under the microscope. Under microscope image, this cross thread looks like this. Okay, so we can see that the green one on the top. Okay, so the green one on the top, so we should put here green. All right. Okay, so the second one is the yellow. Okay, number second is yellow, and the third one will be the red. Okay, red threads. Okay, number 3. Okay, the third one. All right, so we can see that clearly. Okay, so when we focus on the green one, the yellow and also the red will be out of focus. Or out of focus meaning that it's a little bit blur. Okay, when we focus on the yellow one, okay, so the green and also the red one will be out of focus. Okay, same goes to the, we want to focus on the red, another two will be blue. Right, so this is the glue, so don't worry. So this is only glue. Don't forget to draw on our lab report. So when we have to draw on our lab report, okay, make sure that we draw according to the position that we have. For example, here this is the thread number 1. Okay, so number 1 will be, okay, don't forget to label the color and also the position. Okay, number 2 just now is the yellow. Okay, don't worry. Okay, you can do it better than me. Number 2 is a yellow, and number 3 is a red one. Okay, the red. Okay, all right, no need to color, just draw and label and make sure that you put the position number 1, number 2, and number 3. Okay, done. We go to the 1.2 under Fresh Specimen, which is an animals and plant. Under animal, we're going to look at the pond water and simple epithelial cell. B is a plant, which is Elodea, and also onion epidermal. Let's see. Under 1.2 Fresh Specimen, which is an animal, we're going to look at the microorganism in a drop of pond water. So normally, a pond water, they have several types of the microorganism. So we can say that here is, for example, amoeba, Elodea, paramecium, okay, and others that can be found in the figure 1.2 in your lab, which is page number 50. Specimen simple epithelial cell. Put a drop of water on the clean slide. Gently, by using the toothpick, gently scrape the inside of the cheeks with the toothpick and put on the drop of water on the slide. Okay, so we cannot see any of the sample here because it's very thin. Okay, throw it on the biohazard. Okay, we need to put the methylene blue. Okay, methylene blue here is a function to stain or to give color to our epithelial cell because epithelial is too thin. Okay, so take a cover slip and gently put the cover slip on the right angles. Okay, and drop it. Please avoid any scraping of the bubbles and remove the excess water by using a tissue paper. We can use a technique of irrigation technique also, okay, to ensure that the methylene blue will be stained the cell. Okay, so we have to observe under microscope. Under magnification 4 times 10, we can see that simple epithelial cells are quite small, and we cannot see any of the nucleus right here. So we have to move to the high magnification, which is 10. Okay, now we can see very clear. Wow, very nice. Okay, so if you can see that here is the epithelial cell. Okay, so they are smeared very nice. Okay, one by one. Okay, so the size or the shapes of these epithelial cells is irregular shapes. So some of the cells are, this is not very good. Actually, there are abundant of cells. So the good will be something like this, which is a smear, very nice. Okay, individual cell. Okay, we can see that they have the nucleus here, right here. Okay, so under light microscope we cannot see any the organelles. Okay, and only the nucleus. Okay, yeah, we have the magnification for these. Okay, the larger image, you can see that here. Okay, so very nice. Okay, we can see that clearly. Okay, they have the nucleus here, the cell membrane, okay, and also the cytoplasm. Alright, so what about the on the surface of the cell right here? So is it the ribosome? Is it anything that we might know? Alright, so basically it's a bacteria. Wow, okay, so it's a bacteria. So yeah, we have a lot of bacteria. We are the host for other bacteria. Okay, hopefully they are good bacteria. Okay, so from here we can see that the fresh specimen, which is a plant, okay, we can have a Elodea and also the onion. Elodea is we take the green one. Okay, you can see that very clear. Is have the chloroplast. Okay, compared to the onion cell. Okay, onion cell from the microscopic view we cannot see any of the chloroplast or the green plant because is onion bulb. Okay, which is underneath underground of the soil. Okay, for the Elodea cell, we take from the beyond the bean seed channel. Alright. Here is the material that we needed. Put a drop of water. Take a sample, the Elodea sample. Only a small leaf. Normally we advise to take the upper one. And this Elodea is quite thick. Before we put a cover slip, put an iodine to stain the color, to stain the nucleus and also the cell wall. Alright, so under microscope we can see that clearly this is the Elodea with a cytoplasm very clearly. Okay, they are moving. When they are moving, we've got cytoplasmic stream. Okay, next we move to the onion. For the onion, okay, so cut the onion. We need the onion from the inside, not the surface of the onion, just inside. Okay, take a epidermis on the concave side. This video is from the Beyond the Bean Seed channel. Okay, peel the onion. This is a one cell pig. Okay, put the onion on the clean slide and put a drop of water on it. Then don't forget to put the stain iodine, okay, for a better image in our under microscope. Okay, any excess water we have to remove by using a tissue paper. Okay, we can use a method of irrigation of this specimen. Okay, all the specimen we should observe under 40. So before you go to the 40, start with the 4 times, then move to the 10, and last will be 40. Okay, so under light microscope, we can say this is unstained, which is a no color at all. So they're quite what we call colorless. So this is we stain with the TBO or toluene blue O. Okay, we can see that the tissue is a better image. Okay, you can see that they have staining in a better image. Okay, so you can see the nucleus and cell wall. Alright, so this is the onion. Even in the larger image, we can see the cytoplasmic bridges. Okay, this is under 400. Alright, that's all I think. Okay, don't forget to draw the specimen for pond water. Okay, any of the one image from microorganism, you may choose anyone. Okay, draw properly. Okay, and also don't forget to label tick cell also. Okay, and for plant we have the Elodea and also the onion cell. That's the end of our experiment number 1. So don't forget to do our lab report and submit through goals. Thank you very much and Assalamualaikum.